Hello, this is part 3 of the Chapter 11 lecture on genetics for microbiology. So we'll talk about mutations in this one. Mutations are any change in DNA. So let's look at the first slide. The definition of mutation is a change, is a change in any base sequence of DNA. So a lot of people think mutations are always bad, but in fact, they're not always bad. Some mutations can happen and they're silent or they're neutral because there's no change in the protein's function. So let's look at, I'm going to go back a few slides, right? We need to look at the genetic code. And so we see, like in this case, arginine, CGU, CGC, CGA, and CGG all code for arginine. So let's say that the messenger RNA had a U here and mutation happened and that U changed to a C. Okay, that's a change in DNA, right? The DNA ultimately makes the RNA, but it didn't matter because we're still coding for arginine. So in this case, a mutation occurred, right? If we change that third letter anywhere, we would still get arginine. So even if a mutation occurred in that third position, there's nothing that happened to the protein product. It is still arginine that's being coded for. And you can say that about any, right? So any proline, same thing. As long as you have two C's in a row, that third letter can be anything and still code for proline. So there is a wiggle room for having a change in the DNA sequence, but then ultimately nothing happens because we're still coding for that same letter and the same amino acid. So going back to our mutations, that's why they're silent or neutral. We have no idea that they happened because the ultimate protein product, nothing happened to it. Mutations, as we know, can have very bad effects. The protein can be non-functional or less functional. The entire organism could die during the development, depending on how bad the mutation is. If the mutation happened in a very specific spot within a protein that was very important for development, let's say. And then number 3, mutations can be beneficial, right? Because how do you change the organism without changing the DNA? Like ultimately, how a species evolve and change over time is the fact that their DNA does change little by little. So we can actually create maybe improved products that work better, and then that can be passed on to the offspring. So keep in mind that mutations are the basis for evolution, right? You can't change the organism without changing the DNA, which is what I said down here. So mutations are, yes, they are changes in the DNA base sequence, but they can have a series of effects based on like where it happens and what happened to the protein product. So we'll talk about a couple types of mutations. One is going to be a point mutation. Point mutations are just a substitution of a single base. Just one base changes, one letter changes. So again, there's 1, 2, 3, 4. There's 1, 2, 3. There's 3 different things that can happen if you have a change in one letter. So the first thing is what I talked about recently, right? Silent, where you have a mutation. So in this case, it's the letter T right here, right? So it's going to be this A had changed to T, and it didn't matter because the amino acid that we're coding for, valine, still coded for valine, silent. In a missense mutation, this is where you do have a change, right? So in this case here, our codons, remember we read the messenger RNA and codons. We have serine, valine, proline, and tyrosine, and then we have a change, right? So that proline became a tyrosine, I think it is. And this is going to be that iffy one. So is this change enough to change the protein? Is it enough to make the protein dysfunctional? Is it something that might not change the function of the protein? Is it something that might make the protein work better? So we don't know, but all we know is that a missense mutation is that we have one amino acid that had been substituted for another amino acid. So it's kind of like a mistake, missense mistake. You have a different amino acid put into place. The third is going to be something called nonsense. So nonsense is introducing a stop codon where a stop codon should not have been. So let's review. What's a stop codon? In translation, remember we had our ribosome subunits, our big subunit, our small subunit. We had our messenger RNA kind of sandwiched in here. Oops, sorry. Sandwiched in there. And stop codons are supposed to be at the end, right, of the messenger RNA. So somewhere at the end, there's a stop. We know that we begin building the protein at the start codon. So start is always going to be at the beginning, our start codon. And then our stop codon is at the very, very end. And that's what tells the ribosome that this is the end. What if, though, there was a mutation that happened somewhere in here, right? Somewhere in the middle of the gene, and it coded for a stop. This is going to be pretty bad, because what happens then is that the ribosome thinks, oh, that's it. This is the end of the protein. And so we're not going to make the rest of it. So we're going to have a partially made protein. The word truncated is used because truncated means to be shortened, a truncated protein, and probably not functional, right? Probably not. So this is probably the worst kind to have in this base pair substitution mutation. Okay, so a base pair substitution mutation or point mutation is the same thing. It's a substitution of just one base, right? So DNA got a ding that changes the RNA, which may or may not change the amino acid. In this case, it did not, a silent mutation. In this case, it did, right? That change in DNA caused a change in RNA. It coded for a different amino acid. You know, the question mark is, how bad is that change? And then nonsense mutations, we accidentally change the DNA to make a stop codon, which would lead to a truncated or shorter protein. So those are the three kinds of outcomes that can come with if you just change one letter. There is another kind of mutation, which is called a frame shift. So here it is right here, frame shift. So in terms of mutations, the first kind of mutation we have is point mutations. The second kind of mutation we have is a frame shift. Frame shift occurs when you insert or delete one or more of the bases. So in this example here, and honestly, I can't read clearly what's happening here, but there is the wild type, right? And the T and the A. And we see that this was it added in? Oh yeah, the T is added in. I think it was added in. Anyways, it's either you remove. So in this case here, we removed. Oh, sorry, this is the same example. Sorry, this is wild type, so this is normal. And then we can see that we have serine coded for. This is valine. This is proline. This is tyrosine. When you remove a letter, right, the T and A have been removed. Now, all of a sudden, because you're reading in threes, right, 1, 2, 3, 1, 2, 3, 1, 2, 3, the codons, 1, 2, 3. If you remove those two, then the reading frame, you know, those groups of three are all changed, right? So we have serine still, which is normal. And then we have alanine coded for. Then since the shift has happened and the groups of three have moved, then we have leucine. Then we have another one. So we can see that all of the amino acids from the point down have changed. So this is a really big mutation change. Insertions and deletions will drastically change the protein product, because all the amino acids downstream from that point mutation, from the frame shift mutation, whether it's an insertion or a deletion, will get altered. So this is another look, right? So we're altering the codon reading frame by adding or deleting a base. So if this is the normal translation of DNA, where we have our codons here, this is normal, our sequence of amino acids, and then we added one, so this would be called an insertion. If we added one, so we added A, C, G, we added one at the beginning, then we have this frame shift going, and you can see that arginine, it's just completely different. From arginine down, glycine, leucine, histidine, it's completely different. And then if you took one away, same thing happens, right? You have completely different amino acids coded for from the wild type or the normal. So again, much larger effects on the protein because every single amino acid changes after that insertion or deletion. What causes mutations, right? There are two major causes. One is just spontaneous, meaning that there's no outside influence. It can happen with DNA polymerase, right? We talked about DNA polymerase, how it's really good at copying DNA because it has proofreading ability, but it's not perfect, right? It's not a perfect molecule. So DNA polymerase can actually make a change and then it could just stay and then it's there, right? So it's sort of nothing that made the DNA polymerase do that. It just happened because nature isn't a perfect system. The second thing that causes mutations is a specific harmful thing, right? A harmful agent that does cause damage to DNA. And there are three kinds of mutagens. So a mutagen is an agent that either directly or indirectly causes a mutation. These can be physical, chemical, and biological. So physical is like physically changing it. We'll talk about radiation. Like radiation comes in. It is an energy that physically breaks and nicks the DNA molecule. Then we have mutagens that are chemicals. So these are things like, we would call them carcinogens, right? For the chemicals out there in the world that we know are bad for us because they can cause mutations. And the mutations as a disease, cancer is basically the disease of so many mutations building up that your cells no longer function correctly and they start to, divide uncontrollably and they cause cancer. So these can be any of these. You don't need to memorize them, but chemicals can come in, they can go interact with your DNA and they can cause changes to the DNA, which is a mutation. Third, we have biological organisms, so bacteria, viruses. Some viruses we know can cause cancer, so like HPV, right, human papillomavirus, that can cause cervical cancer. What happens in those instances is that the viral DNA gets incorporated into our DNA. We looked at that in chapter 6, how some viruses can actually come in with their DNA and then incorporate their DNA into our DNA and our genome. When they do that, they can cause problems. They can upset a gene that might be necessary for cell cycle regulation, right? So really important for telling the cell when to stop dividing. And so some bacteria and some viruses are biological causes of changes in the DNA. This is an example of a physical cause of mutations, and you do need to know ionizing and non-ionizing mutations. Ionizing is stronger. It is a higher energy form of waves. Think about X-rays, gamma rays. These are things that are like radioactive. Like you don't want to be around them because they'll hurt you, right? Excuse me. So what happens is these X-rays or gamma rays will come in, they will actually nick that and cause a break in the backbone of DNA. And when you come to fix it, it might not be fixed properly. And so you can get, a single-stranded break, you can get double-stranded breaks. So it's really nasty. And this can build up within the cell. And then the cell will become, it'll kill the cell or it will cause cancer eventually. But ionizing radiation is the reason why pregnant women don't want to get X-rays, because they have this developing fetus inside, and any chance that the X-rays could cause those nicks would be really, really bad for the developing fetus. It's also a reason why, the doctors will step out of the room when you're getting an X-ray and you wear that heavy lead vest, because we don't want those X-rays to hit any other target other than like the place they want to take the X-ray from. Non-ionizing radiation is UV, okay? Ultraviolet light. So we're constantly bombarded by UV light. It's in the, sunlight. So what UV light does is it doesn't make those backbone changes, but what happens is if there happens to be two T's next to each other in DNA, they stop binding to the A's across from them, right? They should have A's that they're bound to across from them, but they break that bond and then they form bonds between the two thymines. And that's called a thymine dimer, which is written down here, thymine dimers, when two thymines happen to just bond to each other. This is not normal, and this is a form of mutation, and it does cause a kink in the molecule. Luckily, we have ways to fix mutations when this happens. So if we go out in the sun a lot, right, this is what causes sun cancer, is constant bombardment by UV, constant little changes in the DNA, and sometimes it can overwhelm the repair system, and you can have these mutations kind of stay, in the DNA, they don't get fixed, and then that can cause problems. But both kinds of ionizing radiation, both kinds of these are used to actually kill bacteria. So in the next chapter, we'll talk about ways that we want to kill cells. And when we want to kill cells, we would want to use like X-rays or gamma rays, right? If we want to kill bacteria in like medicine, some product that you're making, like a vaccine, for example, you don't want any bacteria there. You might irradiate it. And then UV light is used a lot in hospitals in like tuberculosis wards. They'll actually shine UV light on in the ducts where they have, the air go through, so that it can kill bacteria that way too. Chemically, you don't have to know this chemical at all, but just know that any chemical that is able to kind of insert itself into the DNA double strand will cause issues. It can cause changes in the base sequence. It can cause a deletion. It can cause an insertion. So most of the time, these chemical mutations will have, or chemical mutagen, right, that causes those mutations, will cause an insertion and deletion. And we talked about how that is a frame shift, and that's really bad for the protein because every single amino acid downstream from that change is different. So the protein is going to be really, really different. Let's talk about fixing the mutation. So in bacteria, we're not going to talk about, there's a whole bunch of ways that eukaryotes, our cells, can fix mutations, but we're not going to address that. There's a couple that you want to know for bacteria, and one is called nucleotide excision repair, which is basically, if you follow this picture here, right, there's a thymine dimer that's produced. We can actually, the helicase enzyme can actually remove a chunk of the damaged section, and then DNA polymerase and ligase will come and fix it. It's a different DNA polymerase, don't worry about it, but you can fix this with the enzymes that are already present, that they monitor DNA, they look for changes in DNA. The other way that bacteria can fix mutations is through an enzyme, and the enzyme is called photolyase. Actually, I should have this bolded because you should know it. Photolyase is, it does something called photoreactivation, and it's simply to remove those thymine dimers. It's called photoreactivation and photolyase because it does require light in order for that enzyme to work. So it's fixing a thymine dimer here. And so bacteria do have ways to get around small mutations. The trick is you want to just hit them with so many mutations that their natural sort of repair mechanisms are overwhelmed and they can't fix all the mutations, and the bacteria will ultimately die. All right, this is the Ames test. This is how we can test for possible mutagens. Let's say there's a new molecule, a new chemical, or there's a chemical existing now, and they want to see, like, is this bad for you? Is this going to cause mutations? So this is what they did. So there is a strain. So the background is there's a strain of Salmonella. So this is bacteria that they use. The Salmonella is unable to grow without histamine. So HIS, you'll see the HIS minus, HIS plus. So Salmonella, that's HIS minus, okay, HIS minus, means that it's been modified to make sure that this bacteria cannot make histidine. It's unable to make that amino acid. It needs that amino acid to live. So if you grow the Salmonella on a plate without histidine, right? Salmonella, it requires histidine. If you grow it without it, most of those bacteria will die, right? But every once in a while, you'll find some colonies because there are natural revertants. What a revertant is that salmonella, just from mutations that happen randomly. Remember, so one thing to note is like mutations, when they occur, it's not directed. The cell doesn't choose where the mutation happens, right? It's a random thing. It just so happens that in this example, a couple of cells had a mutation that put them back into their natural state. So they reverted back to His positive. They can make histidine now and survive without histidine because they can make it themselves. And so this is basically like, okay, this is our control. If we added this bacteria to some liver extracts, it's like a nice nutritive broth, and we plated it without histidine, most of them will die because most of them, they need histidine to live. But some of them will naturally just mutate and survive. When you add a possible mutagen, here we go now, right? So we're adding now this chemical that we don't know about, chemical X. Does chemical X cause mutations? I don't know. So we add it to the salmonella strain that requires histidine, same. And then we plate it. And if you see a lot more revertants, right? A lot more mutations happened that took an HIS minus to form an HIS plus, that means that this chemical here is really active in mutating and changing DNA, and you'll have a lot more revertants because the mutation process has been amplified, right? So the Ames test is used, and this is still used today, is to identify anything that might be mutagenic, potentially carcinogenic chemicals. So a salmonella histidine oxotroph is used to test the strain exposed to a potential mutagen or carcinogen. The number of revertants, right, the number of reversion mutants capable of growing in the absence of the histidine is counted and compared to the number of natural revertants. So between the two, right, what's the difference? What's the difference between the two plates? All we did differently was we added that chemical that we don't know if it causes mutations. And we see that that chemical did allow a lot more of our salmonella to mutate back into the revertant form, right, into the HIS positive form. And so that shows you that this chemical X is a possible mutagen because it does go in and change the DNA, and it amplifies the amount of mutations that do occur. So it's a really simple test, and it's used today even to identify, right, possible mutagens. And then, of course, we want to warn people about those possible mutagens. All right, and that's it for the part 3.